Let's now discuss humoral immunity. I hope you got the point why a laughing Donald Duck is shown here. So, after antigen challenge, B cells also undergo proliferation and differentiation, just like your T cells. Now, there are two ways to activate the B cells. When they are activated by T cells, they're called T-dependent B cells, and they are also called B2 B cells. Those cells that do not depend on T cells for activation are called T-independent or B1 B cells. Activated B cell differentiates into plasma cells and memory B cells. So, memory B cells are secreted by plasma cells. Memory cells do not react right away. They are reserved for later encounters with the same microbe or antigen. These memory cells do not forget, unlike Dory, and they always live long. In this slide, you will see the phases of humoral immune response. Naive B cells recognize antigens. So, naive B cells have IgM and IgD on their surfaces. And under the influence of helper T cell, so this is your T-dependent B cell activation, the B cells are activated to proliferate, giving rise to clonal expansion, and they are also activated to differentiate into antibody secreting plasma cells. Some are activated to undergo isotype switching. We will be discussing this in a while. And then some are differentiated to become high affinity immunoglobulin expressing B cell, and they express IgG. And then some become long-lived memory cells. Now let us see how T-dependent B cells are activated. So here we see a macrophage as our APC, and then we have our T helper cell here, and then finally we have our B cell. Okay, so how does this happen? The microorganism is recognized by the APC, and it is ingested and partially digested, and then the fragments from the microbe bind with the MHC2 to form the MHC2 antigen complex, and then it is presented on the surface of the APC. And then a helper T cell, which is specific for the antigen that is being presented, binds to the complex. After which, the helper T cell then activates an appropriate B cell by releasing this cytokine, this interleukin 2. Now, the interaction between the T helper cell and the B cell causes the B cell to differentiate into plasma cells and memory cells. Activation of CD4 T cells and B cells that we have just discussed happens in a lymph node during a primary immune response. So here Let us review the parts of the lymph node again. So here we have the paracortex, and then here we have the follicle. T cells make contact with the antigen in the T cell zone in the paracortex, while B cells make contact with antigen in the follicle. So the follicle is the B cell zone. Now the CD4 T cells interact with APCs in the paracortex, where the T cells proliferate and differentiate into helper T cell subsets. Some activated helper T cells, such as um, follicle helper T cells, migrate to the border of the follicle. The B cells that have recognized the antigen in the follicle migrate also to the follicle border, where they interact with and receive help from this antigen specific T cell. They are really helpful, hence the name. So the helper T cells bind B cells via this interaction. So it should have this TCR MHC engagement, generating the immunologic synapse that we talked about earlier. The T cells deliver help by interaction of co-stimulatory molecule CD40 present on the B cell and the CD40 ligand that's present in the T helper cell. And T cells also deliver help by secreting cytokines. After the T cell B cell interaction, B cells are stimulated to differentiate and take one of three pathways. First, it can proliferate to form a primary focus. These are the antibody secreting plasma cells that provide the initial IgM antibodies of the primary response. Secondly, it can develop directly to become memory cells that bear IgM. And then finally, it can develop or it can enter the germinal center and undergo one of the most extraordinary differentiation. The B cells that proliferated to form primary focus of IgM producing plasma cells exit the lymph node. Those that re-enter the follicle and join other activated B cells to form a germinal center undergo affinity maturation and class switching. Now here in the dark zone, B cells proliferate and undergo somatic hypermutation. They migrate to sample antigen and seek T cell help in the light zone. Those that successfully compete for T cell help return to the dark zone and proliferate more rapidly and repeat the process. But those that do not receive help will undergo apoptosis. The high affinity class switched B cell clones ultimately differentiate into plasma cells or memory cells and exit the lymph node. B cells expressing IgM and IgD on their surfaces are capable of switching to other classes. This isotype or class switching requires stimulation of the B cell by T helper cells. And it also requires the cytokines produced by the T helper cells. The T helper cells influence the constant region gene to which class switching occurs. So as you can see here, they are switched to become IgG, IgA, and IgE. What about the T-independent activation of B1 B cells? Now, this TI B cells are capable of generating antibodies on antigen stimulation without the need for T cell. Primarily, they protect the body cavities and most particularly the peritoneal cavity. These cells are self-renewing in the periphery, and they secrete mainly IgM antibodies. This is midway between innate and adaptive immunity because antigens bind to innate receptors on the B cells. Here's a question. Why don't ants get sick? The answer is because they have little antibodies. So this slide is actually a segue to the next topic, antibodies. Antibodies are gamma globulins. They are also referred to as immunoglobulins. In humans, there are 5 chemically and physically distinct classes of antibodies: your IgG, IgA, IgM, E, and D. They cannot reach microbes that live inside cells. What they can do is they can bind to microbes before they enter host cells or during passage from infected to uninfected cells. And this is how they act. All antibodies have the same basic 4 polypeptide chain unit. So they have 2 light chains and 2 heavy chains. The class is defined by the heavy chain constant regions. So we have IgM, IgG, IgA, IgD, and IgE. The antigen binds on the FAB fragment. This is the antigen binding fragment. The biological activity is determined by the C regions. It therefore determines the fate of an antigen bound by the antigen binding site. So this is the FAB region, the antigen binding fragment, and this is the FC region, the crystalline fragment. In particular, the C terminal half of the H chain, the FC region or this crystalline fragment, serves other functions which we will be discussing in more detail later. What are the antibody-mediated effector functions? Antibody binding to pathogens or toxins can enhance their inactivation by the following processes. First, there's neutralization. Binding to pathogens or toxins prevent them from binding to cells. Number 2, there's agglutination. The cross-linking of particulate antigens blocks them from binding to cells or tissues. Next, there's opsonization. Binding of Fc receptors on the surface of phagocytic cells induce phagocytosis. And then we have complement activation. When we discussed the complement pathway, we saw that through the classical pathway, we have antigen antibody complexes that will initiate the complement pathway. And then next, we have this antibody-dependent cell-mediated cytotoxicity. Cell-bound antibody binding to Fc receptors on cytotoxic cells, for example, your NK cells, or your macrophages, your neutrophils or eosinophils, activate secretion of cytotoxic proteins that induce apoptosis. And then lastly, there's degranulation. So antigen antibody complexes binding to Fc receptors trigger fusion of this prepackaged secretory granules with the plasma membrane, allowing degranulation. The Fc portion of antibodies here gives them biologic activity. This Fc portion can bind to Fc receptors or FCRs, and these are expressed by many different cell types. We have discussed degranulation of mast cells. So mast cells have these Fc receptors. And then we discussed also opsonization, wherein we saw Fc receptors present on the surfaces of some phagocytes. And then we also discussed previously about this ADCC, or your antibody-dependent cell-mediated cytotoxicity used by your NK cell. So, in addition, we have this Fc receptor present in the syncytial trophoblast. So this is in the placenta, and this helps in the transport of IgG from the maternal blood to the fetal circulation. So we call this the FcRn, and here it is. And then lastly, we have this Fc receptor that functions in the transcytosis of IgA into secretions. So polymeric or poly IgR or polymeric immunoglobulin receptors are expressed on the basal surfaces of epithelial cells of mucosal and secretory tissues. They bind dimers of IgA and also multimers of IgM. And this initiates transcytosis, endocytosis, and transport through a cell to its apical portion. So this is the process responsible for the accumulation of IgA antibodies in bodily secretions. Let's now discuss the different immunoglobulin isotypes. First, we have IgA. So this is responsible for mucosal immunity, and they are usually secreted as a dimer. They are secreted into the lumens of the gastrointestinal and respiratory tracts. They are able to neutralize microbes and toxins. Next, we have the IgD. Before, we do not know the function of IgD, but now we know that it reacts with the bacteria and virus in the upper respiratory tract. It can bind basophils and mast cells, and they also release cytokines. They are secreted as monomers. Then we have the IgE. The function of your IgE is defense against helminthic parasites. But as we will discuss later in hypersensitivity, it's responsible for immediate hypersensitivity. Now, mast cells, as we have discussed a while ago, have Fc receptors. The IgE, which is secreted as a monomer, can bind with these Fc receptors, and this leads to the degranulation of mast cells that cause the allergic reaction. We go to the most versatile isotype of immunoglobulin, IgG. It is also secreted as a monomer, and it has so many functions. It can function in opsonization, so it coats pathogens and prevents them from entering host cells. Now, macrophages also contain Fc receptors to which IgG can bind. So, the antigen that is bound to the IgG is internalized through phagocytosis, and then the microbe is degraded in the phagolysosome, and the microbial peptides are presented on class 2 MHC molecules, and then this is recognized by your T cell. Now, aside from this function, your immune globulin also functions in ADCC, or your antibody dependent cellular cytotoxicity. We have mentioned that a while ago. And it also functions in neonatal immunity. So you have this Fc receptor that facilitates the transport of IgG. And then finally, we have the IgM. This is secreted as a pentamer, and it's the form of antibody that is present in naive B cells. Although it exists as pentamers, the valence is only 5, okay, instead of 10. Now that's because there is crowding, so it cannot accommodate 10 antigens. As with IgA, it also has a J chain for secretion. It is the first antibody to be expressed in mature B cells, as what I have just mentioned. And it is important for the activation of the complement pathway. It appears early after infection and usually not seen upon reinfection. This is a summary of the immunoglobulin isotypes. And indicated here also are the half-lives of these immunoglobulins. The last phase of the immune response is contraction. Shown on this slide is the graph of the CD4 and CD8 T cell response during a primary response to flu virus in the lungs. It initially the response is initially that of expansion and then contraction. Changes in flu virus titer are depicted. So the virus clearance is coincident with increases in CD4 and CD8 cell response, and this occurs between 8 or between 6 days to 10 days after infection. In this particular infection, the CD4 T cell response contracts faster than the CD8 T cell response, which remains evident in the lungs weeks after the peak of infection. Expansion and contraction of the T lymphocyte population are influenced by several growth factors and cytokines. So, the withdrawal of antigen growth factors may play the most important role in reducing cell numbers and results in programmed cell death. Also, the activity of regulatory T cells as well as expression of negative co-stimulatory or what we call as co-inhibitory molecules also contributes to contraction by inhibiting T cell proliferation. And then finally, there is a stimulation of AICD or the activation induced cell death. This also reduces the lymphocyte number, especially in chronic infections. Under humoral immunity, we have the types of immunity. So these are the active and the passive humoral immunity. So in active humoral immunity, antibodies develop after exposure to antigen. So here, the B cells encounter and respond to, to the antigen to produce antibodies. And then in passive humoral immunity, you have the introduction of non-native antibodies. So antibodies are given to the person from another source. The active and passive types of immunity are further divided into two. You have the naturally acquired immunity. So this is natural exposure to antigen. Example is the vertical transmission of antibodies from mother to fetus. And then you have the artificially acquired immunity. Here you have the use of vaccines. Vaccination, it comes from the Latin word vacca, meaning cow. This is in honor of Edward Jenner's work with cowpox inoculation. He is not related to Kylie or Caitlyn. So in 1798, there were milkmaids with mild cowpox immune to smallpox. That's how he started with the idea of giving vaccination. And then Louis Pasteur used cholera on chickens and anthrax on ships. And then in 1885, there was introduction of rabies vaccine by Joseph Meister. Now, putting the two together, we now have naturally acquired immunity. This is obtained in the course of daily life. This is when antigens or pathogens affect the body naturally. So the body generates immune response to this antigens. So immunity may be lifelong, like when you have chicken pox or mumps, or it may be temporary. And then we have the naturally acquired passive immunity. So here, antibodies pass from mother to baby via the placenta or breastfeeding. There's no immune response to antigens. This type of immunity usually lasts for only around weeks to months, and there is protection until the child's immune system has already developed. In artificially acquired immunity, we have artificially acquired active immunity, where the body still generates immune response to antigens, but the antigens were introduced artificially. Example is your immunization. So immunity can be lifelong, like in your polio, or it can be temporary, just like in your tetanus toxoid. And then you have your artificially acquired passive immunity. So here, preformed antibodies are introduced into the body. So the person doesn't generate an immune response. Examples are you have a snake antivenom injection from horses or rabbits. This type of immunity is short-lived, unlike the vaccination. host immune system does not respond to antigens. We have here the kinetics of antibody response. So the concentration and the isotype of serum antibodies following primary and secondary immunization with antigen are shown. The antibody concentrations are plotted on a logarithmic scale. Let's look at the primary response first. It is characterized by first the latent period or the lag phase. This is a period of around 1 to 2 weeks before there is detectable antibodies in the serum. The length depends on the nature of the antigen and of course the sensitivity of the assay. So at this point, your naive B cells are stimulated by antigen, becoming activated and differentiate into antibody secreting cells that produce antibodies that are specific for the antigen. This is the time it takes for the T cells and the B cells to make contact with the antigen. During this period also, there is a proliferation and differentiation of B cells within the primary foci, and there is movement into the germinal center. And then we have the exponential phase. The B cells now of the primary foci release IgM, so the concentration of the antibody in the serum increases exponentially. There's also additional release of both IgM and IgG from those B cells that have migrated into the germinal centers. These are produced by short-lived plasma cells in the lymphoid organs. During the steady state, there is production and degradation of antibody which are balanced. Now, during this declining phase here, towards the end of the primary response, the immune response begins to shut down and the concentration of antibody in serum declines rapidly, but somatically hypermutated receptors have already been produced and memory cells leave the germinal centers. There are also long-lived plasma cells in the bone marrow. Now, what about the second response? This is elicited when the same antigen stimulates the memory B cells. So the lag phase is much shorter and the magnitude of the response is much greater and faster than the primary response. The stimulation of the memory B cells lead to production of greater quantities of specific antibodies. This expanded set of memory B cells bear high affinity receptors available for immediate differentiation to high affinity immunoglobulin G secretion. It is also interesting to note that somatically hypermutated B cells can undergo further hypermutation with additional antigen exposure. The average affinity of antigen-specific antibodies increases through the third and even the fourth immunization with the same antigen. So during the secondary response, you can see that there is this very high magnitude in the concentration of your IgG as compared to your IgM. So if you compare the two, the magnitude of the primary response is smaller than the secondary response. During the primary response, IgM is greater than IgG, but during the secondary response, there is much greater IgG than IgM. And often there is also IgA in immune secretions, and sometimes there is IgE. The affinity of your antibody is lower during the primary response, but during the secondary response, it is higher. So the average affinity is higher, and this is due to your affinity maturation. Primary response is induced by all antigens. Secondary response is produced only by protein antigens. We have finished discussing the adaptive immune response, both cell-mediated and humoral. So we have discussed the features of the adaptive immunity, antigen presentation. We have discussed the development of your lymphocytes, the lymphoid organs, and the tissues. And of course, we have discussed the two types of adaptive immune response. This brings us back to our big picture. So now we understand the big picture more. So we understand what happens when infection sets in. We understand how the innate immunity responds. We understand now the lymphocyte development, the adaptive immune response, your both cellular immunity and humoral immunity. So we now understand the B cells and the T cells, the effector responses, okay, and we also understand about your memory cells. Let's see the bad and the ugly of the immune system. There are three principal ways in which the immune system can fail, resulting in first, hypersensitivity reactions. Here you have a overly sensitive or overactive immune response to an antigen. And then you have your autoimmune reactions. Here, the self is misinterpreted as dangerous, causing an attack on the body's own tissues. So there is breakdown in self-tolerance. Examples are your multiple sclerosis, Crohn's disease, and your rheumatoid arthritis. And then third, you have your immunodeficiencies, primary and secondary. This is there is ineffectivity of the immune response against antigens. Now, you have this transplantation. By identifying the graft as foreign, rejection of a transplant, of a tissue transplant, is an example of the immune system functioning properly. But of course, it poses a challenge in doing tissue and organ transplants. And then you have this immune imbalance. To have a healthy immune response, as in other systems of the body, there should be homeostasis, in that there should be balance between immune pathways that lead to aggression and those requiring inhibition. So you have chronic non-communicable diseases like cardiovascular diseases, insulin resistance, obesity that have been linked to inflammation. Autism, depression, and bipolar disorders are also considered to be chronic inflammation. If this were true, what is the tipping tipping the balance towards the uncontrolled inflammation over immune regulation or homeostasis? Likely explanations include the microbiome, diet, and stress. This have been all this have been shown to have interconnections to the immune, digestive, endocrine, and nervous systems. There is now clear evidence both in mice and humans of a multi-directional interaction between diet, microbiome, and immune function. In particular, it appears that the absence of certain commensal organisms in the gut and modern dietary changes may be linked to tipping of the balance toward uncontrolled inflammation. We will see this in the next slide. There is a proposed role of the microbiome in regulating immune, metabolic, and neurologic function. So, exercise, diet, genotype, microflora, stress, and this antibiotics, the intake of antibiotics, and then you have this body microflora have significant, they all have significant influence on the microbial composition in the gut. These microbes help maintain gut integrity, and this is what we call symbiosis. And this results in balance in the immune, neurologic, and metabolic functions. Now, changes in the diet and other lifestyle factors can lead to disruption of this gut integrity, leading to dysbiosis. This results in immune imbalances that lead to immune overstimulation. So there is chronic inflammation, autoimmunity, and allergic disease. This state results in increased gut permeability and proposed disruptions to other body systems like metabolic disruption, neurologic disruptions. Okay, so this symbiosis is this dysbiosis is believed to contribute to conditions such as type 2 diabetes, inflammatory bowel disease, and other mood disorders. Now, if there is acute inflammation, there is chronic inflammation. Now, what are the causes and consequences of acute inflammation? Chronic inflammation has both infectious and non-infectious causes. But regardless of the cause, chronic inflammatory conditions have common systemic consequences, some of which are related to the effects of inflammatory mediators on metabolism, just like what you have in type 2 diabetes mellitus, and some of which are related to the effects of inflammatory mediators on tissue organization and cell proliferation, like what we have in cancer. Mechanisms behind the association of other disorders may be indirect and are still being investigated. Now, genetic variation also plays a role in both cause and effect. Hypersensitivity reactions are classified on the basis of the principal immunologic mechanism that is responsible for the tissue injury and the disease. We have here the immediate or type 1 hypersensitivity. It is a type of reaction caused by the release of mediators from the mast cells. So the first exposure to the allergen activates this follicular helper T cells so that it stimulates the B cells to form IgE secreting plasma cells. Now this IgE molecules then bind to the Fc receptors present on mast cells and basophils. Now on subsequent exposure, there is cross-linking of the IgE, of the bound IgE, triggering the release of mediators from the mast cells. Now these are vasoactive amines, and this may cause smooth muscle contraction, increased vascular permeability, vasodilation, and platelet aggregation and degranulation. In antibody-mediated or type 2 hypersensitivity, there are antibodies that are directed against cell or tissue antigens. that can damage these cells or tissues or can impair their function. The antibodies can cause disease by first opsonizing cells such as red blood cells, and this will lead to phagocytosis. Second, antibodies can cause disease by inducing inflammation. So here we have immune complexes that can cause disease mainly by inducing this inflammation on the site of deposition. And then on the bottom portion, we have this antibodies. First, we have this antibody against TSH receptor. What is TSH? This is your thyroid stimulating hormone. And this is what happens in Graves' disease. So there is an antibody that stimulates the receptor even without the hormone. You will learn more of this in your thyroid physiology. And then in the middle portion, we have here the events that happen in myasthenia gravis. You have encountered this in your muscle and synapse physiology. There is an antibody that inhibits the binding of acetylcholine to its receptor. And then thirdly, we have this pernicious anemia wherein an antibody inhibits vitamin B12 binding to the intrinsic factor. This therefore prevents vitamin B12 absorption. So, antibody-mediated type 2 hypersensitivity includes blood transfusion reactions, erythroblastosis fetalis, and autoimmune hemolytic anemia. Immune complex mediated or type 3 hypersensitivity. Here, antibodies against soluble antigens in the blood may form complexes with the antigens, and the immune complexes may deposit in blood vessels in various tissues. So this will cause a lot of inflammation and tissue injury. Complement activation initiated by immune complexes. Remember, this is your classical pathway. This produces complement proteins that mediate mast cell degranulation, and they can chemotactically attract neutrophils. So this will stimulate lytic enzymes from neutrophils that will try to phagocytose your C3b coated immune complexes. An example is the Arthus reaction. Okay, this is how it looks like, and it can be induced by insect bites as well as inhalation of fungal or animal protein. Now in this picture, this is caused by a drug given to the patient. It's it caused this Arthus reaction. Okay, so there is deposition of immune complexes in the blood vessels. T-cell mediated or type 4 hypersensitivity. Here we have sensitized T1 helper T cells or type 1 helper T cells. They release cytokines that activate macrophages. Or we have cytotoxic T cells which mediate direct cellular damage. So this is your DTH response or your delayed type hypersensitivity response. Okay, so on the left we have this tuberculin skin test. This test, also known as the MAN2 or PPD test, is carried out by injecting small amounts of purified protein derivative from Mycobacterium tuberculosis. Okay, it's injected intradermally. Okay, and the skin is then examined after about 48 to 72 hours. The skin is examined for redness, induration, Okay, and this is indicative of your type 4 hypersensitivity due to the activation of memory cells. Okay, memory M tuberculosis specific T cells. This is a positive reaction. You will see this in the clinics. This is a list of our learning objectives, and I have also posted this in Canvas. I have divided it into two major parts: WBC or leukocytes, and immunity, which is further divided into two, namely innate and adaptive or acquired immunity. WBCs are components of immunity, so discussion of the different WBCs is incorporated within the immunity lecture. The last part will be application of concepts of the immune response. This last part will only be an introduction, as this will also be studied in your second year and third year classes. Use these learning objectives as a guide in your study. So in summary, those are my favorite words. In summary, we have taken the overview of the immune system. So we discussed about the innate immune system, its components, how it functions, and then we divided the adaptive immune system into your cell-mediated and your humoral immunity. With this, we discussed the special features of the adaptive immune system. We also discussed about the lymphoid organs and tissues and the lymphocytes. And then we discussed about the good and the bad, okay, or the bad and the ugly of your immune system. So these are your hypersensitivity reactions, autoimmunity, transplant rejection, and we also mentioned about chronic inflammation. Okay, so this the last of the series of concept checks. So I hope this will be helpful for you in studying the immune system. I know it's very complicated, and there are so many interactions, but it's really very interesting. These are the references, but you might want to look at Guyton also, because it's in clinical key. You don't have to read these books because they're whole books only on immunology. Here we have two videos that you may view. You can also view other videos um, in YouTube. There are so many videos that you can view. So thank you so much for listening. If you have any questions, please feel free to communicate through Canvas. I the especially through the discussion board under the Introduction to Immunology lecture. Thank you.